Okay, so the goals of this lecture will be that hopefully by the end you'll have a better understanding of the normal appearance of the developing brain, both in the fetal brain and then in the neonatal brain, both preterm and term. And it's important to realize that being able to understand the normal appearance of the developing brain really allows for better detection of abnormalities. And hopefully understand better the sequences for assessing normal brain development and thus detecting an abnormality. And I'll briefly also go over which imaging planes are best for specific structures. And I do want to emphasize here and throughout the lecture that really when you are assessing the appearance of the developing brain on MRI, it's very important to know the gestational age for fetal MRI and the post-conceptional age for evaluation of neonatal brain on MRI. Okay, so here's a question. I was told to put a question at the beginning and at the end. You guys can take out your pads and vote on this, and then I won't go over the answers until the end. So question number 1, when are most primary sulci formed by? A, 22 weeks, B, 26 weeks, C, 28 weeks? I'll give you 30 seconds to enter in your answer. Okay, and then we'll go to question number 2. Oh, okay. You can show the answers, or it doesn't matter, actually. We're going to... okay, question number 2. I'm not sure what he's showing us, but anyways. Which of the following is a true statement? A, the appearance of the brain on T1 and T2 weighted images is similar in fetuses and preterm neonates of the same age. B, the brain appears more mature on T1 and T2 weighted images in fetuses than in preterm neonates of the same age. C, the brain appears less mature on T1 and T2 images in fetuses than in preterm neonates of the same age. Again, you can vote on that. Okay. Can you show me the answers that they gave to, that's question number 1 or question number 2? That's question number 2. Excellent. Okay. So we're going to go on and we'll go over this. Looks like you guys already know most of the answers here. Okay. So in terms of the techniques used to assess normal brain development, really it's similar techniques that are used for fetal and neonatal brain MRI. However, because of the inherent limitations of fetal MRI, namely the small structure and its distance from the coil, as well as the fact that of the fetal brain, and the fact that the fetus is really moving while you're acquiring images, make it much more difficult to perform MR of the fetal brain than of the neonatal brain. Thus, the armamentarium of sequences available to us is actually fewer or less in fetuses. However, in general, we use the same approach when we try to assess the brain in both fetuses and neonates. We look at the morphology and signal intensity of the developing brain, and we typically use in the fetus the T2 weighted images, and in the neonate T1 and T2 weighted images. You can also use 3D volumetric imaging, which can be acquired as a 3D volume in the neonate. In the fetus, you can take two-dimensional images and post-process them into three-dimensional higher resolution images, which I'll show you. We can also assess the microstructure of the developing brain using diffusion imaging. We can use diffusion weighted imaging, which is typically what we do in fetuses because of the limitations, where we can measure ADC values in different areas of the developing brain. And in neonates, we can do diffusion tensor imaging and measure other diffusion parameters such as anisotropy and look at tract-specific measurements. Also, we can assess the metabolic development of the fetal and neonatal brain using MR spectroscopy. And obviously there are other sequences especially available primarily to the neonatal brain, which we will not have time to discuss. So these are the features that really we look at when we're assessing the brain for normal development with MRI in both the fetus and the neonate, and we'll just go through these. First of all, the general approach when you're looking at T1 and T2 weighted images that I have is really to first look at the midline sagittal image, and on that image I particularly look at the corpus callosum to make sure that it's normal in its morphology, that it's fully formed, normal in length, uniform in thickness. I also look at the brainstem for its morphology and its size, as well as the vermis to make sure that it is normally formed. I then use my axial and coronal images to assess the ventricles, especially in the fetus, where I look at the shape of the ventricles, their size, and look at the smoothness of their margins, and also look at the sulcation and cortex then, superficial to the ventricles, and the developing parenchyma, and pay particular attention to the deep gray nuclei. That obviously is important in trying to assess hypoxic ischemic injury in neonates, but also in fetuses, and look at posterior fossa structures on the axial and coronal images. Okay, so we're going to start with the corpus callosum, only to say that really, because it's a midline structure, it's very important to have non-oblique images, and this can be very difficult in young fetuses. The corpus callosum appears as a T2 hypointense structure in the midline above the ventricles. It increases in length and thickness during gestation. However, in general, it has a uniform thickness, relatively uniform thickness throughout, and that's before there's any evidence of myelination within the callosum. In fetuses, because you oftentimes can have oblique images, it's very important not to confuse the corpus callosum with the roof of the ventricular system, because the walls of the ventricle are normally hypointense due to the germinal matrix. And so you want to look for other morphology that will tell you that the corpus callosum has formed or has not formed. And in the neonate, we have here a corrected 30-week gestational age neonate on your left and a term neonate on your right. We oftentimes will use T1 weighted images since we tend to acquire 3D gradient T1 volumetric images, and we can see that it's iso-intense to the unmyelinated supratentorial white matter. Okay, the sulcation and the cortex is obviously something that we can also assess with MRI, and the sulcation in particular is a very dynamic process which occurs in utero and really continues to term and even post-term. And we can see how the appearance of the sylvian fissures, for instance, which are the first sulci to form, are very shallow in a 21-week fetus. We can see on both the axial image and on the coronal images, and at 21 weeks we may also see a parietal occipital sulci, but we shouldn't see any other sulci in the fetal brain. Contrast that to the 23-week fetus, where we have a much more angled appearance of the sylvian fissures on both the axial and coronal images with progressive growth of the anterior and posterior perculum. We can also see here early formation of the calcarine sulcus as this very small indentation on the surface of the brain. Sulcation progresses in utero, and by 24 weeks we can see even further maturation of the sylvian fissures and further maturation of the calcarine sulcus, which is deeper. So it's important that when you're looking at sulcation, not just to look at how many sulci you have and their location, but also to look at their morphology, as they will deepen with increasing gestational age. And by 27 weeks we have many more primary sulci. For instance, we can see the central sulcus here. We can see behind it, this arrow's not working. We can see behind it the post-central sulcus and the pre-central sulcus. We can see the superior temporal sulcus on the coronal and also on the axial plane. And again, notice how the calcarine sulcus has deepened even further. And most primary sulci are complete by 28 weeks gestation. Secondary sulcation will then progress as well. And we can see here a 28-week fetus where compared to the 27-week fetus there are more sulci. And just to contrast that to a 28-week preterm neonate who was born at 27 weeks, to show you that really sulcation is delayed in preterm neonates. And so when you're assessing a preterm neonate, keep that in mind. You can't compare them exactly to the fetus. And if you do, however, you will notice that there is a slight delay in the sulcation pattern in a premature neonate of the same corrected age as a fetus. By 34 weeks, and here's a 34-week neonate, all your primary sulcation is complete, and much of your secondary sulcation, actually nearly all of your secondary sulcation is complete. You can see you have many more sulci. The sulci are much more complex. And then on the images below at term already, you can see that the sulcation pattern is even more complex compared to 34 weeks. And so that's with continuation of sulcation pattern, primarily with tertiary sulcation. You can also take the two-dimensional images that we obtained from fetal MRI and reconstruct them after the images are acquired to obtain higher resolution three-dimensional images. And from that you can model the surface of the fetal brain and look at the formation of areas of curvature on the surface of the brain. And actually with this technique you can see normal hemispheric asymmetries in the formation of the sulci. And also we can see that here in red, we have sulci is detected by autopsy specimen, and in blue, sulci is detected by this 3D technique. And we can see that we can much more better detect sulci at an earlier stage with this 3D technique. We can also assess the developing cortex using this technique, and we can look at the gray matter or the surface area of the cortex and see actually that it increases during gestation. I'll switch now to looking at the parenchyma and white matter. And in the fetus, it's important to realize that the developing parenchyma is actually heterogeneous on the T2 weighted images up until about 28 weeks. And this is really due to the fact that with the T2 weighted images and T1 weighted images, we can see different developmental layers within the fetal brain. In particular, when you look at the fetal brain on T2 weighted images, you can see that there's a hypointense area which represents germinal matrix. Superficial to that, there's a slightly hyperintense area representing the periventricular zone. Superficial to that, we have this more intermediate signal, which represents a combination of the subventricular zone and the intermediate zone, which will grow as it will become, the intermediate zone will grow as it will become the future fetal white matter. And superficial to that, we have a hyperintense area representing the subplate. This is an important transient layer in the fetal brain. It's the first site of synapse formation before the axons make their way onto the cortex, to synapse within the cortex. And then most superficially, we have a T2 hypointense layer representing the developing cortex. And this is seen on coronal images as well as axial and sagittal images, and it disappears by about 28 weeks gestation. We can look at the thickness or the volume of different areas of the developing brain, and using the 3D morphometry technique that I showed you earlier, we can actually show that the subplate, shown here in volume, actually increases between 20 to 26 weeks gestation, and that this ratio of subplate to total volume is roughly constant. Now, we can also look at the signal intensity of the developing parenchyma with our T1 and T2 weighted images, and in general, especially with MR imaging, we are sensitive to changes from myelination. Myelination in general proceeds from a caudal to a cranial direction, dorsal to ventral, and central to peripheral. So these are good general rules to remember. Myelination does begin before term, begins earliest in the cranial nerves and in the spinal cord. And just like other areas of brain development, areas of the brain that are going to serve very primitive functions, such as sensory function and motor function, will undergo maturation first. So you'll see that in myelination, and that those will be the first areas to myelinate before higher cortical functioning areas will myelinate. It's important to realize I'm talking here about myelination, but also when you think about brain development in general, the areas of the brain that are involved in these more primitive functions, such as motor and vision, are the ones that will mature first, whether you're looking at the formation of sulci, whether you're looking at myelination post-birth, or whether you're looking, for instance, at other measures of brain maturation, such as diffusion measurements. And so there's kind of a general pattern of certain areas of the brain maturing at earlier times than other areas, and it's very much related to function. How does myelin appear on MRI? Well, it appears bright on T1 weighted images, primarily due to the formation of cholesterol and glycolipids in the myelin sheath, and it appears dark on T2, primarily related to the decrease in water. And the changes of myelination are earliest seen on the T1 weighted images. Now at 28 weeks, so this is a corrected to 30-week gestational neonate, there's really not much myelin in the supratentorial brain. However, by term, you should see on your T2 weighted images an area of hypointensity within the central area of the posterior limb of the internal capsule, corresponding to the cortical spinal tracts, an ascending up towards the perirolandic region. And similarly, on the T1 weighted images, you should see an area of T1 hypointensity within the posterior limb of the internal capsule, again extending up towards the rolandic cortex. I want to spend some time just talking about the deep gray nuclei, because that's an important area when you're assessing a neonatal brain, especially for the possibility of hypoxic ischemic injury. And on fetal MRI, the deep gray nuclei are relatively isointense to slightly hypointense on T2 relative to the developing intermediate zone, and they're slightly hyperintense relative to the cortex. And you can see that here on the image on your left. However, after about 27 to 28 weeks, the deep gray nuclei become more hypointense and start to become more isointense to the developing cortex. I'm sorry, this mouse keeps jumping. I don't know if there's a way to fix that. And after 28 to 29 weeks, we can also begin to see different areas within the thalamus, and in particular, we can see that the ventrolateral nucleus of the thalamus is hypointense on T2 weighted images and hyperintense on T1 weighted images. And at term, it has a similar appearance, with the ventrolateral nucleus of the thalamus being hypointense on T2 and hyperintense on T1. And the deep gray nuclei are relatively isointense to the cortex on both T1 and T2 weighted images. However, you notice that the thalamus in general is slightly brighter on the T2 weighted images and slightly darker on the T1 weighted images. Again, important to remember the normal appearance of the deep gray nuclei, especially when assessing for hypoxic ischemic injury. In terms of the posterior fossa, we look at the brainstem on sagittal and axial planes and look at the cerebellum in axial and coronal planes. The vermis is important to assess on all three planes. It's important not just to look for the normal morphology of the vermis on the sagittal image, such as the primary vermian fissure, which can oftentimes be seen as early as 21 or 22 weeks gestation, but also to look at the morphology of the vermis and the cerebellum on axial images, and really to see the normal indentation on the dorsal aspect of the fourth ventricle to indicate that indeed your midline tissue is vermis. And the vermis and brainstem and cerebellum, as expected, will grow during gestation, and that's something that can be compared with the literature when you obtain measurements on your fetal MRI. We can also look at the signal intensity, which reflects cellularity and myelination, as myelination does, as I mentioned, begin caudally and progresses cranially, and therefore the brainstem will be myelinated before the supratentorial brain. And we can look at the appearance of the brainstem both in a 29-week fetus, and here we have a 2-week-old X28-week Premie, showing you that we already at this stage start to see hypointense signal within the medulla, within the dorsal pons, within the deep cerebellar nuclei, which you can see here, and also within the flocculus of the cerebellum, a very primitive area of the cerebellum that myelinates earliest, and also within the dorsal midbrain. At term, there's slightly more areas of T2 hypointensity and T1 hyperintensity, again in part reflecting myelination of the deep structures within the brainstem, and in particular we can see the decussation of the superior cerebellar peduncles here within the midbrain as hypointense on T2, reflecting myelination, and hypointense on T1. Okay, we'll now discuss the appearance of the microstructure of the fetal and neonatal brain as assessed with MRI. And we can use diffusion-weighted imaging not just to look qualitatively at the fetal brain and at the neonatal brain and to see if we see areas visibly of reduced diffusion, but we can also use them to obtain quantitative measures. And I strongly recommend that when you are assessing the fetal and neonatal brain with diffusion imaging, that you actually draw regions of interest, for instance, in the deep gray nuclei when you're worried about hypoxic ischemic injury, and you quantitate the ADC values, for instance, and compare them to what is normally observed in those areas. And it turns out that diffusion imaging is another way to look at brain maturation. It reflects the developing microstructure of the brain and in general, something that occurs both in gestation and even after birth. We can see a progressive decline in mean diffusivity or ADC values and a progressive increase in anisotropy with brain maturation or with increasing age. Values of ADC and of anisotropy differ in different brain regions at the same age, and the rates of change will also vary in different brain regions, with areas that are, for instance, affected in more primitive functions, such as motor and vision, as I mentioned, undergoing maturation at a more rapid rate than areas involved in higher cortical functioning. And in fetus, really what we tend to obtain are diffusion-weighted images, as it can be very hard to image the moving fetus, and thus diffusion tensor imaging is still very, very challenging, actually, to obtain in utero. However, when we're assessing the neonatal brain, both preterm and term, we can perform successfully diffusion tensor imaging, and from that we can generate anisotropy maps, we can do color overlay of the different directions, and then from that we can actually get track-specific values of different diffusion measurements, and again, compare with the literature to know what is normal. And so what does the fetal brain look like on ADC maps? Well, here's a 23-week fetus, and you can see how the signal to noise is very diminished for many of the reasons of why fetal MR is limited in general. However, we can see that there actually is lower ADC values within the germinal matrix and within the cortex compared to the developing brain, and we can to the developing parenchyma rather. And we can also see that in the deep gray nuclei there are also relatively lower ADC values compared to the developing supratentorial parenchyma. This pattern stays even at 29 weeks gestation, even though the actual absolute ADC values themselves will change and will progressively decline in most regions of the fetal brain. And this pattern also persists within the term neonate, although already here you can start to see that there is some reduced diffusion in the region of the corticospinal tracts. And in the fetus, as I mentioned, you can measure using region of interest based analysis, the ADC values in different areas of the fetal brain and compare them with normative data. And there are several publications on this in the literature. And again, knowing the gestational age, look at the normal ADC values. Now with neonates, you can obtain diffusion tensor imaging, as I mentioned, and from that you can generate fractional anisotropy maps and even do color overlay. And it's important because with diffusion tensor imaging, when you look at other parameters such as anisotropy, you can see that the brain is not homogeneous on the anisotropy map, and really even though there is no myelin present at this stage in the supratentorial brain, you can see that there is more structured organization of certain tracts within the brain, even at 28 weeks. And you can see that, for instance, within the central tracts or the cortical spinal tracts, within the posterior limbs of the internal capsule, and within the corpus callosum, for instance, compared to the more peripheral white matter, which is largely anisotropic at this age. And as I mentioned, you can also obtain color overlay maps to look at the direction of diffusion. And here, for instance, you can see the corpus callosum bundle. And you can also obtain specific track-based measurements and measure anisotropy values and measure mean diffusivity values and compare them with the literature to know if the brain that you're looking at is normal in its development. Now it turns out that not only does the white matter, the developing white matter, demonstrate anisotropy even before other areas, even before you can detect maturation on T1 and T2 weighted images, but the cortex is also maturing in ways that can really only be seen with diffusion tensor imaging. And in particular, at young gestational ages, we can see that there is anisotropy within the developing cortex, and with increasing development by about 36 weeks, and definitely by term, you should have no anisotropy within your cortex. So here's an anisotropy map showing you that there's high anisotropy within the developing cortex, and by 35 weeks, most of the anisotropy within the cortex is at noise level. Of note, this maturational process of loss of anisotropy within the cortex with brain maturation is something, again, that tends to occur first in the motor areas and visual areas before, for instance, the anterior frontal cortex. And you can look at plots of this and again, measure values, much more for research purposes. Okay, I'm just going to spend the last few minutes talking about biochemical maturation of the brain, and only to say that we can use MR spectroscopy in order to assess the metabolites in vivo. And there are several metabolites that we look for, particularly with our long echo spectroscopy. And in terms of performing spectroscopy in the fetus, it does require a long time to to obtain a single voxel within the brain, and because of that, it's really limited to late gestation fetuses when the fetal head is really engaged in the maternal pelvis and cannot move. And here, for instance, is a single voxel technique using a long TE of 144 milliseconds in a 36-week fetus, showing you that we can already see NAA at this age. We can see the creatine peak and the very high choline peak. And really, the work of Nadine Girard has advanced our understanding of MR spectroscopy in the developing brain, and this is just a slide from her paper in 2006, showing that we can actually look at spectroscopy in the fetal brain even as early as 24 weeks gestation, and throughout gestation we can see a relative change, primarily characterized by an increase in NAA and a relative decrease in choline and myoinositol, as reflective, again, of another measure of normal brain maturation that can be detected in utero. And we can both use long echo spectroscopy as well as short echo spectroscopy, which short echo spectroscopy, although it does let us look at other metabolites, it is, however, more technically challenging within the fetus. Now, it turns out that there isn't just a temporal change in metabolites within the developing brain, but metabolites also vary depending on which area of the brain you're looking at. And for instance, here are spectroscopic values from a preterm neonate in the thalamus, basal ganglia and frontal white matter compared to a term neonate, and you can see how the NAA is much higher in the developing thalamus as compared to the frontal white matter. And you can see that between 30 weeks and 40 weeks, there's some change with increasing NAA and relative decline in choline within the thalamus, even more markedly in the maturation of the basal ganglia, and not much change in the frontal white matter, as there aren't many maturational changes detected at this stage by MR spectroscopy. So in conclusion, the appearance of the fetal and neonatal brain on MRI differs depending on the gestational age and on the post-conceptional age, and familiarity with the normal appearance at different ages is very important to be able to identify abnormalities. So we're going to go to our questions again. If you can hopefully just answer these, and we'll go over the answers. When are most primary sulci formed by? A, 22 weeks, B, 26 weeks, C, 28 weeks. Okay, can we see the answers? Excellent. Okay, and most primary sulci are formed by 28 weeks. The last primary sulci will form at about 33 weeks. And number 2, which of the following is a true statement? A, the appearance of the brain on T1 and T2 images is similar in fetuses and preterm neonates of the same age. B, the brain appears more mature on T1 and T2 images in fetuses than in preterm neonates of the same age. Or C, the brain appears less mature on T1 and T2 images in fetuses than in preterm neonates of the same age. Okay, and why don't we show the answers of that? Excellent, and that's very important to realize because, one of the motivations for doing fetal MRI is to better understand how the brain is supposed to develop, and with fetal MRI we are realizing that the brain development ex utero is not quite the same as the brain development at the same age in utero. So hopefully that will give us better insight into some of the developmental abnormalities that occur in the preterm brain. So I want to thank you for your attention.